Welcome to Inside the Hive.tv podcast, the show that takes you into the world of bees. I'm Dr. Umberto Boncristiani. In today's episode, I speak with Dr. Adrian Fisher II, assistant professor in the School of Life Sciences at Arizona State University. In this interview, we discuss the important topic of how the EPA currently regulates new pesticides entering in the market, the potential flaws in the regulatory processes focusing on pollinator protection, and the proposed solution Dr. Fisher and other authors published in the prestigious journal BioSciences. Before we jump into the conversation, I would like to thank the team of Vita Bee Health. Vita Bee Health is the only company dedicated to bee health and is always bringing you solutions to help bees and beekeepers around the world. Thanks Vita for sponsoring this channel. Please check them out, link in the description of this video. And now, I present to you Dr. Adrian Fisher II. Hello everyone. Today, it is with great pleasure that I speak with Dr. Adrian Fisher. Adrian is an assistant professor in the School of Life Sciences at Arizona State University and was kind enough to donate his time to talk to us today about one of his publications that really got my attention, entitled Breaking the Cycle, Reforming Pesticide Regulation to Protect Pollinators. And it's with great pleasure that I'm bringing you guys today Dr. Adrian Fisher. Hey, Adrian, how are you?
I'm good. Thank you very much for having me on.
Oh, it is a pleasure. It's been a while that your publication got my attention, but I never had the opportunity to take the time. And now I think all the energies of the world come together and we're finally making this. I'm very excited to have you here. Let's start this conversation talking a little bit about you. I want to introduce you to my audience. So my people at home might not know you, and they really like to know the researchers that are working hard every day to bring new information to the public so we can always improve our lives. So where are you from? What's your background? Tell me a little bit about you, man.
Yeah, definitely. So I was born and raised in Southern California, and I just grew up loving animals. I wanted to work with them someday, but I know there's some students in attendance. When I got to university level, I still had that love for animals, but I wasn't really sure what to do with it. And I took an entomology course. I did well in it, and the professor for that course invited me to participate in some of her research. And she just happened to study honeybees and native pollinators and how some of their foraging behaviors differ at regional agricultural systems. So that's really how I got started on my current trajectory. And from there, I would, by the way, my undergrad institution was Cal Poly Pomona. From there, I went on to Texas A&M University, where I took more of a toxicological angle. I looked at the impact of different pesticides on drones or male honeybee fertility and worker survival. And there was a big emphasis on realism in terms of the concentrations and doses that we were subjecting honeybees to, are reflective of what they would actually experience in the field. And based off of those experiments, we found negative impacts both on drone fertility and on worker survival. I then continued from there to Arizona State University as a postdoc, and I expanded further on fungicide and climate change impacts on honeybee health. And that kind of theme of realism carried over with me, where I wanted to, again, subject honeybees to realistic concentrations, but also capture the colony component, which is really lacking in a lot of pesticide studies that are very heavily allocated to laboratory conditions. But anyway, so I was able to continue here at ASU, and now as faculty, and I'm still interested in a lot of questions regarding environmental stressors and their impacts on pollinators.
Wow, congratulations for your career, now a faculty member, now building your own team, and you're going to be express more of your ideas more freely, I guess, depending if you get the funding for. So good luck with you and your team. So only good things. The Inside Out TV society is cheering for you and your team. So, Adrian, pesticides are always, in my view, a controversial topic. I have to say, every time I try to talk about, I face some kind of problems. Some researchers don't want to come and talk live with me or a recording session, and you are one of the ones that agreed upon to do that. And so I first of all want to appreciate your I would say courage to come in and talk about pesticides and your work. And I want to start by asking, why do you decide to put this piece of work together? The title is very enticing to me. It's very powerful. Breaking the Cycle, Reforming Pesticide Regulation to Protect Pollinators. Pesticide regulation is another thing that is very problematic. This is a subject, in my view, that we need to address very carefully, because if we, on one side, we want a tool that works. So we have from one side, we have three sides of this story, right? We have the farmers, we have the pesticide companies, and we have the environment and everybody else, I would say. Because the pesticides companies, they want to make money. They want to make a product that have value for the farmer, that's going to make the farmer have value for us, basically, to produce food and quantity and quality. But those three players, they kind of sometimes get confused about what value is. I'll put this way. And I want to know your thoughts about that. How can you make sure we have a system that's not going to hurt the farmers? At the same time, it's not going to make the industry do not want to produce other products or bring other products to help the farmers. And in the middle of that, we, the consumers of food, is going to be looking at that or being poisoned with something that, how do you see this whole dynamic in your view?
Right. Yeah. And as you kind of alluded to in your question, it is a very complex and at times complicated issue. I find, and this comes in consultation with colleagues, but also in just my own experiences, that there is a dearth of information and communication and dialogue between these different parties that you mentioned. There's always a clear objective communicated between growers and the manufacturers of these products, and then us as researchers that are more interested on the impacts of these products on the environment. So having a clear understanding of the fact that growers are trying to protect their bottom line, trying to maintain a certain level of crop productivity, and pesticide manufacturers are often maybe a bit more, I don't want to say biased, but angled in that direction, where they're thinking about what's going to best serve this interest in producing a certain level of crops. But what gets left on the back burner is, what is the fallout? How do non-target organisms get affected? And even questions about the persistence of some of these pesticide products in the environment are not clear. And that's kind of where we, the researchers that are more interested in those environmental questions, come into the picture. And I think currently, unfortunately, it's almost a antagonistic relationship under the surface, in a sense, where those of us, and granted, I'm coming from the bias of being in that third category where I'm more interested in those environmental questions, where we are, from their perspective, maybe attacking their best method to protect their crops, and the economic side for the pesticide manufacturers. So really breaking down those barriers and coming together on shared interests and goals. Obviously, farmers and pesticide manufacturers do not want to get rid of honeybees or other pollinators. These are essential for those agro ecosystems. So maybe starting on the common goals and kind of benefits of all of our work would be the best starting point, and then we can flesh out where we differ and find better harmony at resolving some of those distinctions between the different categories.
Yeah, it is a complicated one. When I talk with farmers, sometimes they complain that if you take the tools they have, most of them don't want to hurt bees or pollinators or anybody. But at the same time, they need to put, they need to be, it's a business. They need to keep the business healthy. And the complaint that I got the most is not even like, they say, some of them tell me like this: I don't mind, give me another tool, or if you're going to get rid of this tool, make sure you don't allow importation of food from people that you allow to have that kind of tools, because then become an unfair competition. You're just hurting me and all my competition outside, other countries that do not have a strict regulation. It's going to come up and have a quantity, a product that is gonna hurt them economically. So the way I feel that most of the farmers, they want better solutions. I think they already know from experience in the field that those chemicals are not exactly the best choice of work. And now, after I don't know how many 40 years now, with so many data we have, I think it is a fact now. I don't think there is any discussion anymore about how those pesticides also hurt, other organisms that people was not thinking about at the time, or non-target organisms. So it is tricky. It is tricky, and I'm always trying to educate myself and bring the experts like you to discuss those topics with me. Can we go and to your paper and go to the figures, and you give a little overview about what you put together there, because it is beautiful.
Certainly.
So the title is Breaking the Cycle, Reforming Pesticide Regulation to Protect Pollinators. Can you tell me a little bit about the authors here? I want to give credit to everybody.
Yes. So this paper really did come out, I guess, as one of the few silver linings of the pandemic. So many of the co-authors on this study, we really met for the first time and started our collaboration as a result of the fact that the Entomological Society of America, or ESA, the biggest entomological organization here in North America, they switched in 2020 from their typical in-person format for their annual meetings to online. And at the time, I was a postdoc, and my postdoctoral mentor, who's actually also one of the authors, John Harrison, and we put together this symposium idea where we wanted to invite researchers that looked at different pollinator species and pesticide impacts on them. And because of this switch from the in-person to online format, we were able to expand our net, so to speak, and invite international researchers. They could record themselves and upload it to the website, so they wouldn't have to be burdened with the travel expenses and other logistical matters. So again, we were able to get a wider representation with more international flavors contributing to the overall discussion. And when we conducted our symposium, we were all struck by the fact that despite the fact that we were looking at different study organisms, different pesticide products in different regions, we all were finding common themes in that these approved pesticide products were having detrimental impacts on the variety of different pollinator species that we studied. And these were impacts that were not necessarily mortality or some of the more striking die-offs that you might see reported in the media, or that really is the standard for a lot of regulatory agencies, but numerous different types of sublethal effects and other impacts that, again, would not be reflected in some of those standard regulatory assessments. So this really, or rather I should say, that symposium and the subsequent discussion really stirred this interest among us to do something about it and to put together our findings and address what is the root cause. And in our discussions comparing the Environmental Protection Agency, the EPA here in the U.S., with the EFSA, which is the regulatory agency for the European Union, and perspectives from Brazil and China and other locations that are also big pesticide manufacturers and users, we kind of deciphered that one of the common themes were inadequacies in how a lot of these pesticides are tested and ultimately approved for usage, and the fallout that occurs, and the fact that this process just repeats itself with a new product that takes the place of previously banned products. And it really was an issue that we locked in on, and we suggested solutions to try to break that cycle to provide longer term sustainable solutions.
Sustainable solution is the key words here. Adrian, can you tell us how the regulatory works now? Can you give a little overview about how things work right now, and I'll ask you questions based on whatever I understand from it. And because I am from the field of honeybees, but I don't consider myself a specialist in pesticide in any case. So that's why I'm gonna ask you maybe what do we need? I have here all the pictures. If you need this picture, we can open up.
Sure. Yeah, I think the figure would be helpful for explaining this.
Okay, so how I make it bigger? Think I need to take this. Just 1 second. I need to come back. Give me 1 second. I'm going to get there. I'm going to do this instead. All right. I think now it's better because I can have full control of the picture. Here we go.
There we go.
So, there is A and B. A is the way is currently the regulatory agencies use, and B. Can you give us a little overview about what you're seeing here?
Definitely. So we can start with side A, which is how things currently run. And it's not explicitly stated on that figure, but I would argue that there are three main parties involved. There are the pesticide manufacturers, which are essential for the very first step, where that box is testing pesticides.
New pesticides, right here. Okay.
Right. So they're the ones manufacturing these products, formulating different products to be put out on the market. And then starting with the next step where we progress to toxicity tests under laboratory conditions, this is where these products are then submitted to the EPA, which is the main regulatory agency here in the U.S. And for the European Union, that would be the EFSA, and then there are other analogous organizations and other international locations. But the EPA would take these products and typically test honeybees as the model system. And these are going to be honeybees that are removed from the field and placed in cages in the lab, and then given different concentrations, either through tactile or contact exposure or through oral or feeding exposure. And those concentrations are going to be based off of label rates or other standard issue application settings. And on the basis of those toxicity tests, there's usually one of two follow-up procedures. Well, actually, I should mention, perhaps as a pre-step to those two options, oftentimes mathematical models are employed to try to predict or anticipate any potential impacts should this pesticide be approved and applied in field conditions. So,
but no tests.
I'm sorry, what was that?
But no test in the field. So let's keep moving. So we are here. New pesticide, toxicity test in laboratory conditions, topical or oral in adult bees, in larvae. Just right. Three. Do they use in other organisms or only honeybees?
Yes, that is one of the main flaws. Honeybees are used as the go-to model for pollinators.
Yeah, we have a problem there, right?
Absolutely.
Because honeybees are very, very different from almost everybody else out there. All the other bees, the majority of the species are not social. We have many other organisms. So to test something in an organism that's completely different from the rest of the organisms, that could be problematic.
Exactly. Honeybees are more of an exception than the rule when it comes to life history and biology of many bee species.
All right. So they do those laboratory work, and then they apply a mathematical model and try to extrapolate what's going to happen in the field. Is that a fair assumption here?
Correct. Yes, that's perfectly described.
Okay. So they estimate the exposure from the model, and then what do?
Should both the model and the laboratory tests indicate maybe some issue, they may opt for perhaps first a semi-field trial where they have hives in cages out in the field. and maybe from there progress to more of a proper field test where the hives are out in the open and there's some degree of regulation of their exposure. However, that is more of an optimistic expectation. These field and even semi-field tests tend to, even if there are issues detected, not be conducted, mainly because the main argument is expenses involved and just some logistical issues with maintaining adequate sample sizes to properly vet what pesticides might be doing to bees in field conditions. So oftentimes, and the larger green arrow is trying to indicate this, you might see a direct progression from the initial laboratory test and the modeling test to just risk assessment. And this incorporates a more legislative angle where the economic turnout and expectations are also evaluated, in addition to some considerations for environmental impact.
Oh, so that's the part that is tricky, is the capitalism part of the system, because this costs a lot of money. Well, let's discuss this first. Sure. How that is realistic? I mean, you're putting just one component here, right? The new pesticide. This is not combined with any other pesticides that the bees are exposed to, or any other conditions here. This is very well controlled, and even the chemicals here is the pure chemical, not the real product that is combined also with adjuvants and things like that. Am I correct?
Those are excellent points. Yes, it's usually just the active ingredient, not the entire formulation. As you mentioned, the adjuvants or surfactants and other products that can be incorporated in the final overall formulation.
People don't know that? I'm asking, looks very basic to me. So if that's not going to express the realistic conditions the bee is going to pass through in nature, in the real world, why do they do that?
That's a great question. And quite honestly, I don't really have a solid answer for you. I can say that perhaps the root of the issue goes all the way back to the first step with the pesticide manufacturers, and not to point fingers, because ultimately I do want to encourage cooperation between all of these parties. But
yes,
and this is often reflected on the labels of many of these pesticide products. You only really see the active ingredients listed, and the other inert ingredients are you don't really get a detailed list of what those are, if any indication of what they are. And apparently this is an acceptable format or standard for the EPA, that the assumption is that those other components are not going to exert any kind of toxicity effect or outcome for bees, and it's just the active ingredients that are assumed to potentially have some kind of impact.
Wow. And then from here, you can go straight to the risk management, or if something happened here, it's not mandatory, apparently, based on those arrows here.
Exactly.
So somebody can come here and argue and find a way to go straight to risk management instead of go to a potential field test with no cages.
Correct. And actually, my understanding is that's what typically is done. It pretty much, that larger green arrow is the typical pattern where we go straight from the modeling exposure test to risk management assessments.
So in the current pesticide regulation process, the great majority of the pesticides are approved directly from here, right? They estimate in the first they do the laboratory test, which is, let's talk about this a little bit, because even with honeybees, there are they are the exception instead of the rule of the organisms out there. But even that test is very short period of time, right?
Absolutely.
It's just a couple of days, but that's not realistic. Well, exposure in a real life situation. Honeybees are exposed to pesticides almost every day, their whole life, their whole adult life. The adult bees do not reproduce, so they don't see the reproduction of the hive or any other parameters that can compromise the whole the whole situation here. Is that a fair estimation here?
Absolutely. And there's also the angle of the persistence of residues of these chemicals within the hive itself, which, as you touched on, can lend to continual exposure.
So a lot of flaws from here. I don't know about the mathematical models, but if you start a mathematical model with something that have a lot of flaws, I know for a fact that the result is not going to be good. So, and based on what I'm seeing here, from here go straight to the risk management that we're going to talk in a little bit, with very few of those chemicals passing through some sort of field trials that have always their own flaws in each of them.
Correct.
Wow. I have problems with that because I do those tests myself. Sometimes I spend some money and you can see your own water, your all surroundings around you. You can have a lot of that crap. I'm sorry with my language, but that's the reality. But okay, let's move on. So when they do the field tests, do you know how much bees, how much colonies, how that works?
Right. So, kind of touching on something I mentioned earlier, this aspect of field realism has been a major part of my own studies. And I usually opt for as many hives as I can get, usually 40 plus. The EPA and other regulatory agencies argue that they would need hundreds of colonies or at least 50 plus colonies to really effectively flesh out any potential effects of exposure to pesticides. So, as I mentioned, it is very rare, if ever, that that final stage, that these field tests are conducted, mainly due to those concerns regarding cost and time investment and really being able to monitor these colonies long-term for any potential impacts of exposure.
No doubt, it is a lot of money, that for sure. Those hives and the people involved and how long the study is going to be involved, all those chemical tests are very costly. So it's a hard one. And I see why this arrow is right here most of the time. Boom. Okay, so risk management. So what do they do here?
So I alluded to this earlier, but this is more so the legislative side, where they consider legal ramifications, economic impacts of approving and applying this particular pesticide. And part of it is assessing how effective it will be in controlling whatever the pest target is, and thus contributing to maximized yield for whatever the crop or crop systems of interest for which this pesticide will be approved.
So it's basically a short economic, that's what right here. Yeah, short-term economics. It's just to make sure the product's good. It's supposed to bring some value for the end consumer of the product. Basically, they want to make sure that if you apply this amount, this field, you're gonna get extra amount of fruit comparing it with if you do not apply something like that.
That's right. I don't want to say that there is no scientific analysis considered. That obviously is a soul for consultation, but it is more so the economic legislative side of this overall approval process.
And then is approved. So, and how can you approve? Without knowing if it's spread in the air, if it go to the water, and if it reach other places? That's it. They approved, and then people can use. That's the way it's done now.
Yes. So one of the solutions, or rather, I should say, one of the list of solutions that we came up with, and that'll be reflected in Model B, was that aspect of post-approval monitoring, but also just deciphering the overall impact, answering those questions of persistence and where residues will end up, and not just in the crop system, but as residues in the soil and water and some of the things you mentioned, the air or dust dispersal as well. And those really are not incorporated in the current model as illustrated by Figure 8.
So, and then people start to use here, and then the researchers identify problems, and then the pesticide is banned. Sometimes,
Right.
I would say sometimes, and then the cycle started. So, and that's the cycle that you are talking about in the paper. That's the cycle that you want to break.
Correct.
Okay, so let's talk about the suggestions you give here, because they much more interesting in my view. Okay, new pesticides in your way to see how things is supposed to be done. Give us a little overview here.
Right. So proceeding from that first step, which would more or less still be the same, where we have some new pesticide formulation or product that is developed by these manufacturers. We kind of touched on it in our discussion of in Figure A, but one of the things we would suggest is to look at the formulation, because that is what is actually applied in the field, not just the active ingredients. And other researchers have found effects of some of these so-called benign or inert ingredients on pollinator health. So taking into account the full breadth of effects that these formulations might induce. In addition to that, we want to eliminate the short-term aspect of the laboratory setting, where I don't think I specified, but typically those laboratory tests are only looking for 48 hours, and they're strictly looking for mortality. So our suggestion would be for these laboratory tests to expand the time period to get a better sense of realistic exposure duration and any long-term effects of exposure, but also to monitor sublethal impacts of exposure, not just whether the bee is killed or dies within a certain time frame.
I recently spoke with Dr. Scott McCord from Cornell, and he was telling me that it was a very interesting way to put it. You don't measure, it's only insects dying, but you also need to measure insects that are not being born. They're having problems reproducing. So this is not death. It's just how can I put this? Stop reproduction somehow, which is also part of perhaps the reduction of population that we see around. Is that correct? Because everything is based on being dying, but nobody's looking at bees that are not being, coming to life, put this way.
Right. So in other words, fitness effects.
Yes, fitness. That's the word that I was looking for.
Right. Yes, and I think Scott is spot on in pointing that out. And we do have some discussion, particularly with our field trials, but even you can imagine these laboratory tests could look at reproductives and any outcomes of exposure for queens and drones or males and females of non-honeybee species.
So here in your model, I'm going to call your model, but all the authors and things, I see honeybees, and I'm assuming this is a bumblebee, which is also a social organism. There is any other organisms that are not social here that should be included? I don't know. Because this is another exception, I would say. Yes,
and that's a good point. The bumblebee we put in to represent all non-honeybees. Because as I mentioned before, in Model A, honeybees are the only pollinator. They are used as a model for pollinators in general. So we didn't mean to suggest that we would only look at social species. We definitely would want to look at solitary species, and especially since their lifestyles are more typical for bees in general and most other pollinators. But yeah, the bumblebee was more of a representation of that non-honeybee aspect that we want to incorporate in these assessments.
What also is here, I would assume, is because it's another organism that is easy to test. People grow those colonies in companies, and it's easy to access them in laboratory in the right quantities to do the test that we need, correct?
Yes, that's a good point as well. And actually, to that effect, while they haven't implemented it yet, the EFSA, which is kind of the European analog to the EPA, they are a bit further along in discussions of incorporating bumblebees in these tests as well.
Any other solitary bees that we can produce in laboratory conditions and the quantities that we need to do the kind of tests? Just to have the three of them, two socials, one one more. I need one more to be happier.
Right. So there has been some success with orchid bees, with family Megachilidae, but that is admittedly a bit of a challenge. It is difficult to maintain a lot of these solitary species in laboratory conditions. These are often soil nesters, and they have other particular life history traits that do not necessarily translate well to laboratory settings, as we see with some of these more social species.
I know, and I'm aware of that. I'm just poking you because it is a challenge. I want to make clear for the people at home that, at the same time that we would like to have a system to be, to test everything we can possibly test, but there is costs. There are some organisms out there that we don't have the amount of quantity to do it. So become a challenge. That's why we need a whole cycle of potential ways to see the problem, to get as much information that we can get, to get better solutions from that. So, okay, so we discussed the chronic metastasis. We put more bumblebees. We're thinking about other extend the amount of time the exposure is going to be in a laboratory condition, so we can get more data from this. So we did our estimations here. So what happens now?
So from here, unlike how it's currently run, our suggestion is that if both the laboratory tests and the follow-up mathematical modeling suggest some major issue, that the pesticide would be sent back to square one, where it would need to be reformulated and seriously modified to reflect a safer product, potentially for pollinators and other non-target organisms. Conversely, as the green arrow is indicating, if those two initial tests indicate that this might be a relatively safe product for non-target organisms, then we would further expand by, and we kind of touched on this, looking at potential effects on even more diverse pollinators and other non-target organisms that could realistically affect or expect exposure to these chemicals, with an emphasis on deciphering sublethal effects. Because as I mentioned before, there's this really a sole focus on mortality, but reductions in biological fitness, as we were discussing, or behavioral alterations, or impacts on learning and memory, and other important characteristics of these various organisms are also important considerations that can affect populations and overall survival of various species.
Just for the people at home to understand this very clear, it's not only about killing the bees. It's also about to let the bees have their whole reproductive system working properly. So we can have more bees, they can reproduce themselves in the rate that the nature needs them for. So when we're talking about sublethal effects, we're talking about memory problems, right? Some bees going to have memory problems and not going to be able to go outside, memorize and do everything they need to do to go back home with the right food, or not even come back home because they don't have the memory system. So the brain is not working properly. Therefore, the whole colony in that aspect is going to suffer. And the colony suffering, there's going to be less and less offspring from that specific colony. What other sublethal effects we're talking about here, other than memory loss?
Yeah, so there might be some degree of dulling of senses. They may not have as acute of a sense of smell or vision. And that could also lead to, as you were describing, getting lost more often, or not being able to find resources as effectively in the environment. Something that we've seen in our own research is some bees that are exposed to these pesticides are weaker. They're physically weaker. So they're not able to carry as heavy of a load of pollen or nectar, and not able to fly as efficiently as bees that are healthier, not exposed to these chemicals.
Wow. So they can also be outcompete by other organisms too. So
right.
A lot of, and this is not only one or the other. We're talking about the combination, everything happening at the same time, right?
Absolutely.
So it's a big hit. Even though the bees don't die, they got knocked down. Basically, that's the appropriate word. And then it's hard to work when you're knocked down.
Right. I mean, if you can think of it as to maybe make an analogy with human well-being, poisons can make us sick or disable us in some way, but not outright kill us. And we can see something similar in bees where they're sick or disabled in some way, but they're not dead. So that still would negatively impact their colony, or if it's an individual or solitary species, their own well-being.
That's a great way to put with the human examples. Yeah, it's hard when we get sick to compete, to do the things that we need to do.
Right.
All right. What changed with the pesticide applications in outdoor cages? Is still the same, or in this case, we're going to use the whole product with the whole combination of products that end up the final product?
Right. So I would say that probably the biggest takeaway at this part of the figure is that in concept, the two next steps are the same: these outdoor cages and then the more unbounded field tests, as what is described in process A, or how it currently runs. But we would recommend that these be required. It's not a matter of being able to bypass them and jump straight to risk management. But,
oh yeah, I remember now. Here,
right.
This is not mandatory here. So
exactly.
Okay, so the big difference here that from A, which is the way people are doing right now, the great majority go straight to risk management. But in your point of view, we should make it mandatory to pass to these two other tests. Correct?
Exactly. Definitely.
Cool. So yeah, I see improvement. I will vote B. I will vote B. So what else now? So we're here. Anything change in the risk management and the short-term economics, or anything right here, or can I go straight to the other one?
Right. So as I mentioned before, in terms of those two follow-up tests, they are in concept similar. They would allow for a more proper ecological understanding of the impact of pesticides along with other stressors, and just in general give us a better sense of how bees in field realistic conditions are experiencing pressure from these different chemicals. With regard to risk management, we would still see many of the same elements, those economic and legislative considerations, but now those parties would be more informed by a more thorough scientific process examining the impact of these proposed products on pollinator health, and also from the lens of more diverse species covered, not just the honeybee, but other bee species, whether they're social or solitary, and even other pollinators, that non-bee pollinators, that could be incorporated in this overall system.
Right. With more data, the approved pesticides much more likely will be safer, not only for the bees, but for everybody else out there. And now there is something different here, if I remember correctly. Here, we didn't have anything. And now we have something called post-approved monitoring. What is that about?
Yes. So as the name suggests, this would be continuing to assess the potential impacts, long-term and otherwise, of pesticides, even after approval. And we took the approach of what is known as pharmacovigilance, and this is in the human model, assessing long-term impacts of approved drugs on human well-being. We can see developmental rates in children and adult outcomes from children that may be exposed to different products in their youth. And the idea is kind of similar in that even after these chemicals have initially been deemed to be safe or relatively safe and are approved, we can examine if there are any longer-term effects of exposure, both directly for pollinators, but also do these chemicals linger in the environment, and does this aspect also pose a risk for various non-target organisms? As we know, issues of runoff where residues of these chemicals end up in aquatic environments can be devastating for various species that are not pollinators, but they're still organisms that are impacted by our current practices with pesticide approval. So the impact on some of these other organisms that are peripheral to agricultural systems, as well as those that are directly in these agricultural systems, would be taken into account in this overall post-approval monitoring process.
That makes sense to me. And even for the companies producing those pesticides, I would say would have some sort of effect on that. they don't need to do it again over and over. If you do it the first time better, you might not need to keep redoing it, unless that's what they want, which is a subject for another podcast, I guess. It's a whole different discussion. Yeah. Cool. So, make a lot of sense to me. Where are we with this? Do you think is coming? I know that New York just banned partially neonicotinoids in corn, and it's going to be a big reduction of almost more than 90% because of the reports from Cornell, Dr. Scott McCart put together. And I know that we're recording this on April 26, 2024. I know that yesterday Vermont passed through the Senate a similar bill, and it's going to the government. So I'm curious to see if you know anything about that, if there is any other efforts going in other places, people are talking about any other efforts from regulatory people to perhaps to implement this cycle B here, let's put this way, the good one, the one that I vote for, that is more robust to generate the pesticides, safer pesticides. Do you know anything?
Yeah, so unfortunately, I probably don't have too much good news to share. Those measures you described, I think, are helpful, but I would view them, and I mean no disrespect to saying this, but I would view them more like band-aids in a sense, and that it's kind of still falling in line with cycle or figure A, where you're seeing these bans of certain products, but then you expect replacement products to be produced and run through the cycle and eventually approved, and probably on the other end, those replacements will be found to have these adverse effects on non-target organisms. And right now, we're still set on this cyclical process of these new products coming in, eventually being banned, or at least falling into disuse and being replaced by new products. So I would argue that to truly see some changes, it would have to happen more towards the front end. And by that, I mean both on the part of the manufacturers, but maybe even more so on the part of the regulatory agencies, the EPA and the EFSA and others. And I would argue that currently the EFSA is a bit further along than we are here in the US with the EPA, and that they are having more discussions and pushing towards, at the very least, incorporating more diverse pollinators to test, even if the tests are still have some of those flaws with just examining mortality within a 48-hour window or so.
All right, so we have a long way to go, I guess. What I can do here is I'm going to try to bring some people from EPA to talk here in the podcast. I'm also in contact with people in Europe, so we can have discussions and see we can have more information directly from Europe, what they're doing over there. I think that's the way I can contribute here, so people at home can be informed and make their own decisions when they vote or see any sort of policy changes out there. Adrian, I wanna switch a little bit and talk about perhaps your research. You mentioned some very interesting things to me over there about pesticides and drones. Can you elaborate on that a little bit?
Certainly. So I guess I can start with a little bit about my background as a graduate student at Texas A&M. That's really where I got started on pesticide research. So probably the one of the big themes or questions I was considering were looking at some maybe poorly studied pesticides that kind of fly under the radar, even among bee researchers, and their impacts on bee health. And one alarming thing we found, and actually this came from a study that was published some time ago, where wax and other hive products were chemically analyzed to just give a sense of what residues were present in those products. And we were really alarmed by not only the high number of different pesticide products that are in wax and pollen and even honey, but also in some cases how highly concentrated they were in the samples that were analyzed for that study. So on that basis, we wanted to see if some of the chemicals or pesticides that were highly concentrated in wax the rearing environment for honeybees had any impact on development in drones. And in drones, which are male honeybees, their reproductive development is mainly completed during the juvenile stage, so during pupal development. So even before they emerge as adults, they're pretty much well equipped with their reproductive organs and sperm cells and such. So we suspected that if a lot of wax and other high components are having these high levels of chemicals, then drones are experiencing this topical exposure with transfer of these residues into their tissues, and that might impact or compromise their reproductive fitness. So we conducted this experiment where we sprayed new drone frames with known concentrations of some of those highly detected pesticides and reared drones in those environments, and we were able to rear them into adulthood and then collect them and assess their sperm viability. And we did find that in the presence of those pesticides, they had significant reductions in sperm viability and presumably reproductive competence relative to drones that were reared in a more clean environment.
So that's going again to the whole thing here with that model. Now you're saying that bees were not dying, but the reproductive system is being affected.
Exactly. So those drones, just on the surface, looked like normal drones. They were not different in terms of their survival or overall lifespan, but they were compromised in their reproductive ability. And as you might imagine, queens that may be mated with those drones would be compromised in their own reproductive fitness. And so any colonies that those queens then went ahead and established would maybe have fewer workers or die sooner or have a shorter overall lifespan than colonies that were headed by queens that had mated with healthier drones.
People at home, my fellow beekeepers out there, to remember queen issues. This is another part of queen issues. When they're bad mating with bad drones, bad everything. And keep that in mind when we're doing your risk assessment for your own application of products out there, that sometimes you are shooting yourself in the foot. Put this way. Right. Any other project you are involved right now that you can share with us a little bit?
Definitely. If I could just make one final note, since I really like that you addressed a point to beekeepers, also relevant to beekeepers. The three main products that we tested were actually miticides that are applied by beekeepers to control Varroa. So these miticides that are intended to protect bees are in turn actually negatively impacting bees, at least in terms of fertility and perhaps other ways that we don't completely understand. So that point about being careful about what we do in our management practices is definitely well placed.
Yes, mites are problematic. We need to find ways to deal with mites, but we need, cannot kill our bees to kill the mites. Please don't kill the bees in the process. Basically, that's the message, I guess.
Right.
Adrian, tell me about other projects that you're involved, because I'm really curious now. This is very interesting, and I'm very likely going to make a video about this paper. If you send me the reference to me, it's another paper that a very good candidate to become a video here in the channel.
Oh, yeah, absolutely. I could do that. But anyway, just speak to something that has been in the works more recently. A big area of emphasis has been looking at field realistic effects of fungicide exposure. And I'll just say for the audience, I presume that maybe you could probably decipher what a fungicide is just based on the name. It's a pesticide that's specifically tailored to control fungal pests and pathogens. But as a group, fungicides generally have flown under the radar, both in the sense of from the regulatory perspective and those tests that we described in those figures. Fungicides rarely kill bees at the same rate that you might see with insecticides and other chemicals. So believe it or not, fungicides were historically considered to be bee safe. And even some of the products had a label that said safe for bees or bee safe because of the EPA tests where fungicides didn't kill bees in that short time period. But a number of researchers, and myself included, have found negative effects. And more recently, I've looked at a fungicide that is known by the trade name Pristine. And this until literally maybe a couple of years ago, was the most widely used fungicide on almonds and other major bee pollinators crops. So we were actually able to collect pollen samples from bees that were in almond orchards and chemically tested that pollen, and found out the approximate concentrations that they were experiencing in terms of exposure to pristine and other products. So we then used those detected concentrations as a basis for replicating field tests where we had colonies out in our apiary, and we designated certain colonies for different concentrations of the fungicide. And our goal was to, again, not only test exposure effects in these field conditions, but also long-term effects of exposure. And in short, we found that after weeks and weeks, if not months of exposure, colonies that were even exposed to tiny concentrations below the field range had reduced population levels. They had altered foraging behaviors. They tended to collect more pollen. And kind of the backdrop to this is we suspect that this fungicide impairs protein digestion. And so the bees were compensating by trying to bring in more protein, more pollen. Bees that were exposed to this chemical, to this fungicide, also had reduced learning and memory performance. So they were not able to learn as effectively and remember that information. And this might say something about their ability to forage effectively or make it back to the hive if they're out in the environment. And last but not least, we saw that these bees have reduced lifespans as well. So although it didn't kill them outright, they ultimately survived about maybe 20%. Their lifespan rather was about 20% shorter than bees that were reared in a cleaner control environment.
Wow, the fungicides are in my radar too, especially after Reed Johnson and Ohio State University. He came up with those synergistic effects with some miticides, with fungicides, and the power that was, I was shocked by, 1 + 1 is not 2, it's 1,000 in those cases. And the capability of combinations of chemicals interact with each other in a synergistic effect and amplify the lethality to the bees to 1,000 I was shocked to see the possibility and really concerned. Did you test any synergistic effects? Do you work with that kind of research in your laboratory?
Right. I don't have anything currently, but as a graduate student, I did actually look at synergistic effects between fungicides. And we did also see somewhat of a similar outcome where fungicides could amplify the toxicity of each other as opposed to a single application of an individual fungicide. And part of the backdrop to this is just the mechanism of action of a lot of the active ingredients in these products, where they're unlike insecticides, which often target neuronal networks and other nervous system functions. These fungicides are mainly tailored to target cellular machinery, intermembrane proteins of the mitochondria, and other subcellular processes. And one big target are cytochrome P450s, which are these enzymes that actually confer detoxification capabilities. And when these proteins are impaired, that's part of the reason why other chemicals that are combined with these fungicides can be enhanced in their lethality or toxicity for bees.
Yeah, it is a complicated world, I guess. After I learned everything, all of this, Adrian, with synergistic effects and all those, and being in the field, and I know from the reality how pesticides are sprayed and how people sometimes don't follow the label too much, and and we know, we have data that those pesticides end up in many different places where they're not supposed to. And knowing that they are very reactive with other chemicals, I start to be so paranoid with, we have medications that we use. I don't think there is anybody that have ever tested those medications interaction with the pesticides that end up in our food. How can you sleep at night? All those combinations, and sometimes I see people with cancer now. It's very likely one of those interactions that nobody can track. I don't know. What are your thoughts about that? How can we protect ourselves from these potential synergistic effects that nobody ever gonna test? And we know it happens. We know those chemicals are interacting with each other, finding each other. Oh man, that's a concern of mine. What are your thoughts on that?
Yeah, so it is somewhat of a nightmare scenario in a sense, because even just going to the grocery store, the fruits and vegetables you buy have some pesticides on them, some traces at the very least, and not to mention the plastics and microplastics and other such potentially toxic substances. So it really is a very unexplored frontier, and you can imagine, as you were alluding to, how impactful it can be for our health, and yet how very little we know about how these different elements might be interacting with each other and contributing to some of these ailments that we are seeing in our own species, not to mention other organisms. So I honestly, I can't even begin to suggest how we would even go about testing that, but I would hope that, at least in terms of the contribution of pesticides to that overall stew of other chemicals that we experience, some of these procedures where we are being a little bit more careful about what we approve and how we monitor their persistence and impacts on other organisms might at least contribute to the reduction of the potential impact of pesticides on other organisms, including our own health, relative to some of these other chemicals that we are exposed to.
The reality is that the big experiment happened in the population, I would say. The big experiment really happened in the population, and there are nobody tracking what's going on in those possible interactions. That is a big concern of mine and my dogs and all the people that I love. And I have a problem with that because, when you ingest a chemical, a new medicine that is you taking it, you choose to do it, is your own risk. There are ways to. This is not going to end up in anybody's food or.
Yeah.
But pesticides, it's somebody's tools that is ending up in my water, in the food that I eat. And how can I protect myself against those things? That's why one of the biggest problem why I have problem with pesticides and want to try to find other ways to help the farmers to replace from those chemicals, because I am convinced that these chemicals are hurting much more than we can possibly track.
Absolutely. I would have to share that sentiment. I don't think we've even really scratched the surface on the full breadth of the impacts that these chemicals are having on humans and a variety of other organisms.
So that being said, I want to ask you maybe one last question, because there are RNAi technology, something that I worked before, and some of the big companies are moving to that direction to have double-strand RNA, which is a molecule, but it's I would say there is the potential to be much, much safer and very, very specific to the organism they're targeting. And that could be a revolution in agriculture if they really find a way to scale and make sure it works the way it's supposed to. Do you know much about this field and what's going on with that?
Right. I admittedly am not super familiar, but I would have to echo the sentiment that this really could be, if we could get a good grasp on it, a very effective replacement, mainly due to that high level of specificity that these pesticides, despite their intention, do not provide. I know that their use, anytime you're talking about genetic tampering or tools, that they're, from a social perspective, could be a lot of controversy. There's already a lot of concern about GMOs and other, genetic procedures we engage in now. But it does, granted, from the outside of
Sure.
looking at it, it does seem like a very promising potential alternative to our current practices.
From when I think about the synergistic effect, those combinations that is happening everywhere, and we cannot track the big problems. I really feel that RNA could be a good way to go. But only the research is going to tell us. Oh, Adrian, do you have any message to people at home? If they wanted to contact you, where they should go, and the email, people want to interact with the researchers.
Yes, I'm a big proponent of openness and dialogue with general communities, especially since our research on pollinators has direct application for society. So for anyone that wants to find out more about my research and what it means to the food you eat or anything or any other aspect of concern, I'm more than welcome to receive emails or other forms of communication and talk to you more about it.
I will put the information about his lab and how to find Dr. Fisher at the description of this video. Adrian, in the name of the InsideTheHive.tv community, I want to thank you for your time. I want to thank you for the great work you do. Researchers are heroes that that bring new information all the time for society, so society can improve in every single aspect. So in the name of the community, thank you very much.
Thank you very much for having me. I appreciate it.